Now we will discuss about the collagen. Overview. Collagen. Collagen is most abundant protein in the human body and major structural component of extracellular matrix. Collagen is basically an abundant protein in the human body, and it is also a structural component of extracellular matrix or ECM. What it does, collagen provides tensile strength, organization, and support to tissues heavily modified after translation. Post-translational modifications. Major collagen types and locations. Collagen has 4 types: type 1, type 2, type 3. Type 3 is also known as reticulin, and type 4. So type 1 collagen. Type 1 is found in bone, skin, tendon, dentin, fascia, cornea, and during late wound repair. Primary role of type 1 collagen is tensile strength and structural support. Clinical note: decreased production in osteogenesis imperfecta type 1. Type 1 collagen is decreased. It has decreased production in the osteogenesis imperfecta type 1. Type 2 collagen. Type 2 collagen is found in the cartilage, hyaline cartilage, vitreous body, nucleus pulposus. And the primary role of type 2 collagen is cartilage structure and resilience. Type 3 collagen is also known as reticulin, and type 3 collagen is found in the skin, blood vessels, uterus, fetal tissue, and early wound repair. And primary role of type 3 collagen is reticular fibers in expandable tissues. Clinical note: it is deficient in vasculars, Ehlers Danlos syndrome. Type 3 collagen is deficient in vascular Ehlers Danlos syndrome. Type 4 collagen. Type 4 under the floor. So type 4 collagen is found in the basement membrane and basal lamina. For example, glomerulus, cochlea, and lens. Primary role of type 4 collagen is network forming collagen under epithelial layers. Clinical note: type 4 collagen, it is defective in the Alport syndrome. So how will you remember that? So Alport has basement, so type 4 under the floor. So type 4 collagen is defective in Alport syndrome, and it is targeted by autoantibodies in Goodpasture syndrome. Okay. Cells involved and wound healing. Fibroblasts synthesize collagen precursors and carry out most collagen production. Myofibroblasts secrete collagen during proliferative stage and mediate wound contraction. Molecular structure. Alpha chains follow repeating sequence GLYXY or GlyXY. Alpha chains follow repeating sequence that is known as GlyXY. Glycine occurs at very third residues, and it is essential for the tight packing of triple helix. X and Y positions are often proline and lysine. Y is frequently hydroxylated. Collagen synthesis steps. So there are various steps of collagen synthesis. One is translation, hydroxylation, glycosylation, exocytosis, triple helix formation, proteolytic processing, self-assembly and fibril formation, and cross-linking. Translation: synthesis of pre-pro-collagen alpha chains on the rough endoplasmic reticulum. So it happened in the translation process. In hydroxylation, proline and lysine residues are hydroxylated to hydroxyproline and hydroxylysine. It requires vitamin C. So deficiency of vitamin C causes scurvy, weak collagen, and poor wound healing. That happened in the scurvy. Glycosylation. Hydroxylysine residues are glycosylated to aid folding and stability. Triple helix formation. triple helix formation. 3 pro-alpha chains from pro-collagen triple helix stabilized by hydrogen and disulfide bonds. Failure of triple helix formation leads to osteogenesis imperfecta, that is defective helix, and leads to brittle bones. Exocytosis. Procollagen is secreted into the extracellular space. Proteolytic processing. Cleavage of N and C terminal propeptides produces tropocollagen, that is insoluble. Self-assembly and fibril formation. Tropocollagen molecule align and assemble into collagen fibrils. Tropocollagen molecules aligned and assemble into collagen fibrils. Cross-linking. Lysyl oxidase. It forms covalent lysine hydroxylation that cross-links stabilizing fibers. Cross-linking that increases with age and defects in cross-linking leads to Menkes disease, that is impaired copper transport. Key enzymes and cofactors. Prolyl and lysyl hydroxylases, they require vitamin C for hydroxylation. Prolyl and lysyl, these are enzymes, they require vitamin C for hydroxylation. Lysyl oxidase, it is copper dependent enzyme for cross-link formation. Disulfide bond formation, it is important for procollagen stability and triple helix assembly. Clinical correlation. Scurvy, it is vitamin C deficiency and that leads to impaired hydroxylation. Fragile collagen, bleeding, poor wound healing. Osteogenesis imperfecta, mutations affecting triple helix formation of type 1 collagen that leads to brittle bones. Vascular Ehlers Danlos syndrome, that is type 3 collagen deficiency that leads to fragile blood vessels and tissues. Menkes disease. Menkes disease is actually defective copper transport. It is due to defective copper transport. Reduced lysyl oxidase enzyme activity. In Menkes disease, lysyl oxidase enzyme activity is reduced, that leads to poor cross-linking and connective tissue defects. Alport syndrome, that is type 4 collagen defect, and that is defect in the type 4 collagen. Basement membrane dysfunction. So Alport syndrome basically defect in impact kidney and ear. Goodpasture syndrome. In Goodpasture syndrome, autoantibodies are against type 4 collagen. That basically Goodpasture syndrome basically damage in lungs and kidney. That leads to lung and kidney damage.